What's up, everybody? I hope you're doing well today. Today is going to be a fun video, but I had to make some sacrifices. And so one of the questions I see a lot that is asked, but then also one that apparently is the most searched is how do carnivorous plants work? And so I thought I would do something really fun, and I would open up some pictures and talk through how carnivorous plants work. And so this will be a part one to a larger series where I'll go through each carnivorous plant and explain to you just what's happening and how it works. So right off the bat, we're going to look at the beautiful psittacina that we have talked about a lot here, if you've been following. And so the psittacina is a pitcher plant, American pitcher plant, and it part of the Sarracenia family. And the way that this one catches prey is that prey enters through a hole, and then it has to do these false windows that makes the bug or fish go down, and then it goes down the pitcher and it gets stuck. And so that is what I'm going to show you. Let me switch over to my right hand. Here I have a cross section of this psittacina, and so this, what you're looking at, would be this part right here. And so this right here would be the closest area to the base of the plant, and this would be the here we go, and this would be the little bulbous part right here. So right here is the opening, and so that's right here would be the opening. Now the insect or fish, if this gets dipped in the water, would come down here and would go into this opening, and right here on the edges, or excuse me, on the sides, which would be right here, you can see it on the external side of it, are those little false windows. And so what happens is the bug sees the light coming in through these windows, and it thinks, okay, the exit is this way. And so it comes down and it goes around this little hoop, and it's a little hard to see, but right here are a bunch of downward facing hairs that point down towards the base of the pitcher plant, or the pitcher. And what happens is this bug goes down here, it doesn't understand the insidiousness of all this, and so it continues to follow those windows. But what happens is once it gets to this point, it can't go back because of these downward hairs. And so here is where the carnivory occurs. There's going to be fluid in there with enzymes, and it's going to start to break down that prey that cannot escape because of these little hairs that are pointing down. So that is how a psittacina works. And now let's talk about purpurea. So next we have a cross section of a purpurea, which is also a pitcher plant in the Sarracenia family. Now you'll notice its hairs are at the lid and they are facing down. A little hard to see here, but they're there. But you can see it here on the whole one here. Now purpurea works a little bit differently, and so it's going to have fluid in it, and it wants fluid in it, and it's going to have beneficial bacteria and enzymes that help, or bugs that help break down the prey, and then it gets its nutrients that way. So you'll see here that it's more of a bowl shape. So this would be, if I could, it would be more like that, as you can see here. And so there isn't as much, oops, let me see if I can pick this back up. There isn't as much going on as far as that nefarious hairs pointing down, holding prey in. This is more of a reliant on a sort of a pool or a drowning situation. And so what happens is the bug comes up here and it starts getting nectar, and it starts trying to get a little bit further down, and it gets on these hairs, which breaks up a lot of insects have the ability to grip on to things, and so that helps break that up, and the hairs are pointing down, so they fall in. And once they fall in, the death pool, if you will either drown the insect, or it will slowly, unfortunately, start to eat the insect alive. And so that is what happens there. And so that is the cross section of a purpurea, and it kind of works just in that same realm. And so what ends up happening is there are cells along the side of the walls that will ingest or absorb rather the nutrients that are broke down in sort of that pool of ickiness. All right, so next we have a Sarracenia. Now I went ahead and cut this one off because unfortunately even though this is a new pitcher, some slugs had gotten a hold of it, or some caterpillars rather, and they had chewed it all to bits. And so I'm gonna have to go looking for that here in a little bit. But very similar to the purpurea. Now I'm gonna show you a regular one, so one that's whole rather. And so here we have the lip that's helped keep rainwater out, but there is nectar all around the edge here that gets the insect drunk. And let me see if I can get the focus, there we go. And so what happens is the insect gets the nectar, partakes, and then it falls in, and then that's where we have sort of this pitfall mechanism again. The insect comes in here, and about right here, there's going to be fluid in here. And the pitcher's releasing enzymes into that fluid that breaks down that breaks down that insect using a thing called chitinase. It breaks it down and it absorbs the nutrients into the cell walls on either side of the pitcher that then provides nutrients for the plant. All right, next we're going to talk about Drosera. Now, Drosera has a couple different types of mechanisms. There's an action mechanism where the plant actually moves around the prey, and then you have sort of a passive mechanism, which you'll have here with this filiformis. Now, the filiformis, Droseras are also known as sundews, and they have dew all over the leaves, hence their name, and it's very sticky. I don't know if you can see that, but very sticky. Now, this is produced, it's called mucilage, and it's produced by sugars that the plant uses based off of photosynthesis. And what is happening is the insect goes unsuspectingly to try to get some water, thinking this is water. It gets stuck, and then once it gets stuck, it sort of wiggles around and it further gets ensnared, and the plant will start producing enzymes to break down the outer exoskeleton. And so that is what this plant does, but it does not move. It sort of just stays there, and it starts collecting nutrients. You can see here all these little fungus gnats or flies that have been caught, and that's what happened. Now, banana is going to do the same thing. We have a beautiful, probably staghorn, it looks like here. It's going to be doing the same thing, but it takes on a more teamwork approach, and so it's producing a ton of these large splaying branching leaves along with all its other leaves, and it makes quite a large mass, and that mass is going to be working sort of to catch as much prey as possible. Now, there are a couple different Droseras that actually move, like a Brahmanii and a Capensis. I'm going to show you these Capensis. Now, what's cool about a Capensis is not only does it act in the same way the filiformis or the banana that I just showed you, but it will also curl around the prey. So as the prey moves, this will start to curl around and completely engulf in a circular form the prey, and then it will take the nutrients from that. And the reason why it does that is to get as much surface area on the insect as possible. And so I wish I had an example of that, but it doesn't happen in real time like a Venus flytrap, but it will happen over a period of a couple hours, maybe an hour or so. It'll start to cool around the animal. So if you have a time-lapse camera, you can see it. But Brahmanii has trigger hairs on them, which is interesting, on the very edges that do move in real time, and they flip forward, hoping, let's say this was a Brahmanii, which it is not, but insect is crawling by and here it barely touches the outside, it will try to flick it so it gets stuck in the middle, and it gets stuck that way. But I don't have any of those right now, but that is how sundews catch it. You either have a passive like a filiformis or a banana, there's a whole bunch of spatulata, rotundifolia, there's a bunch of them. But then you also have active movements like a Brahmanii or a Capensis. All right, finally we're going to talk about the most famous carnivorous plant, the Venus flytrap, and how it catches its prey. So the Venus flytrap has little trigger hairs. You can see they're denoted by these little darker spots here. Those trigger hairs, they are placed all around the interior of the trap. There's a action potential that occurs here. When the trigger hair is hit, it releases calcium that floods into different parts of the trap that sort of causes an imbalance in the cells, which causes the closure of the trap. Now, these trigger hairs have to be hit twice. It will release some calcium the first time, but the second time it releases enough to sort of tip the scales on that trap. And what will happen is that trap will close in real time. Let's see if I can actually, now you don't normally want to do this, but let's see if I can. Oh my gosh, it is hard to use. Let's see if I can get, there we go. So you don't normally want to do this too much because the traps only have about 2 to 3 times and it uses a lot of energy, but for purposes of education here, we'll go ahead and do that. Now, at this point, the carnivory has not occurred. Right now, the plant has just closed the traps just to prepare itself. What's going to happen is if I was an insect and I was in there and I would continue to wiggle around trying to find a way out, I'm going to keep hitting those trigger hairs. And those trigger hairs are going to keep triggering more and more calcium to release. And that calcium is going to flood in and keep tightening and tightening and tightening that trap. Once it reaches a certain point of being completely sealed, it will continue to release, or it will start to release enzymes, and that is when the breakdown starts to actually occur. Now, how are carnivorous plants able to be carnivorous? And that all comes down to an evolutionary process using chitinase. So carnivorous plants are able to be carnivorous because millions of years ago, they had a protein, they still do, but they had a protein in them called chitinase. And this was used to fight chitin, which is an active ingredient in fungus. So it was a natural fungicide. Now, because the soil was poor and nutrient depleted, the plant realized through evolution that chitin is also the same thing that makes up the exoskeletons of insects. And so it repurposed that chitinase to break down the cell or the wall structure of insects, and in that way started the process along to start getting the nutrients from inside the insect. And so these plants release that chitinase to break down the chitin that is in the insect's exoskeleton and eat its prey.